Yo, what up, Sci fam? It's your boy Nickel, and we're gonna learn and dig into the structure of a chromosome. Now, as you learn about genetics and different aspects of cells, you learn a lot of vocabulary terms related to DNA. And so let's just unravel the mystery of chromosomes. I use that word as a pun. But anyway, we'll get there. DNA in a cell has a molecular structure that is called a double helix because it's like a ladder that has been twisted. So a lot of times it'll be like a twisted ladder. So you can see here the blue strand and the orange strand. These are two strands that make up this strand of DNA. And if I were to be a bit more accurate here, is that actually one strand is connected to the other strand in the middle, and so they're not actually just connected as one single thing, but they actually pair up. One side has one of the base pairs for DNA, and the other side has the complementary base pair. So if one side has the letter A as a base pair, the other side would have a letter T as a base pair. If one side had a letter C as a base pair, the other one would have a G, because G and C are complementary to each other, and A and T are complementary to each other. So if the top strand had a T, the bottom strand would have an A. If the top had a C, the bottom would have a G. Okay, cool. So all those little lines I drew are actually A's, T's, C's, and G's in different orders. And this molecular structure, like I said, this is called a double helix. And thanks to Rosalind Franklin for taking the X-ray chromatography to discover that. And Watson and Crick also have been credited with creating the model of this double helix. And this is also known as, or at least a double helix is basically a twisted ladder. So I like to add that in there just as a reminder of what that is. All right, so DNA in a cell is typically in this double helical shape, the twisted ladder shape. And most of the time, DNA in our cell is in the form of this stuff that we call chromatin. And so chromatin, if we look at this cell that we have here, okay, this here would be called the cell membrane. You can see all these other organelles in there, but this big brown organelle, the one that I made the biggest just so that we could really emphasize what's going on inside of it, this is the nucleus. All right, inside of our nucleus, we have this spaghetti string stuff that's all kind of relaxed and hanging out. It's almost like if you thought about like a ball of yarn, like a ball of yarn that's like a ton of yarn all in a spool. Well, imagine just taking one of those pieces of yarn. That right there would be considered chromatin. And if you could even unravel that one strand into the little pieces, that's going to be our DNA. So inside of the nucleus, we have all of this like stringy looking stuff, and this is called chromatin. So let's label that. The DNA is inside of the nucleus, DNA, but it's in the form of chromatin. Okay, but as we learn about mitosis, we learn about these things right here called chromosomes. And so chromatin actually makes up a chromosome, and a chromatin is made out of DNA. So the DNA strands all basically combine together to make chromatin. And so we're using this little model of yarn. So we have DNA, which makes chromatin. Chromatin makes this bigger thing. This bigger thing, the spool of yarn, would be considered a chromosome. So right before the cell divides in mitosis, or even in other process of cell division known as meiosis, the cell condenses, or actually not the cell, but actually let's say that before the cell divides, the DNA condenses. And even more specifically, let's not even say DNA. Let's say that the chromatin, sorry if you had to erase a bunch there, wow. Right before the cell divides, the chromatin is going to condense and become more compact, and it's going to fold or spool itself, like I showed up with my awesomely drawn spool of yarn here. Right before the cell divides, the chromatin condenses and folds into chromosomes. So again, we're still talking about the same stuff. DNA makes chromatin, chromatin makes chromosomes, just like a ball of yarn is made out of tiny little threads, but even when you unspool the yarn, there's one string, and even that string can be unwound into several smaller strands. So we have chromosomes, which are made of chromatin. Chromatin is made out of DNA. And when we see chromosomes, when they're usually drawn, they're drawn like what I have down here. Now this one's a little bit extra fancy because what I'm showing here is the chromosome structure. And on a chromosome, different segments of the chromosome, if we could unwind that, that'd be a ton of chromatin all wound up and packaged into that little part of that chromosome there. And this chromatin actually contains the information for the different traits. So this little area would be a trait, this little area would be a trait, this area would be a trait, and so on. So I want to get rid of all this extra clutter though, because what we're going to do is we're going to look at some of the vocabulary terms that are associated with looking at an overall chromosome. You see, they're usually in this like X shape because if you look, they're actually duplicates of each other. And in mitosis, when the cell divides, we need to have an exact copy in each of the two cells. So therefore, one of these sides will go into one cell and one side will go into the new cell that is created, or I guess not new, but one goes into one of the copies, the other goes into the other copy, as the one cell makes two cells in mitosis. So we need to make sure we have an exact copy of each so that each new cell ends up with the same DNA as the original cell had. So each of these things right here, since they're exact copies of each other, the term that we use is called a sister. Here comes another chroma word. These are chromatids, so sister chromatids. And the sister chromatids are connected together with this thing in the middle. This thing in the middle is like a little piece of masking tape called a centromere. And so in mitosis, the spindle fibers pull these two parts away from each other by ripping apart the centromere. So the two sister chromatids are pulled apart from each other during mitosis so that each new cell or each cell ends up with the same amount of DNA with the same genes. This overall thing though, as we've already said a million times, is our chromosome. So there you go, a little bit of an understanding as to how a chromosome is actually made out of chromatin, and chromatin is just stuff that's been a little bit more compacted, and that stuff is DNA. So DNA makes chromatin, chromatin makes chromosomes, and now you know the rest of the story.